All right, I want to turn our conversation just even if briefly to NK cells and non-viral approaches to genetic engineering, which can be combined effectively, though there are some challenges. And so, uh, but my first question I'd like to pose to you, as it applies to Celularity, and again, you and your professional experience, what are the advantages of NK cells for genetic engineering from your perspective?
A good question, and I have to admit I'm biased because since our discovery of the sort of unique type of NK cell that can be found in the placenta sometimes in cord blood, but it can be derived from the hematopoietic population and then resident in the organ itself. NK cells offer certain advantages in genetic engineering because they have an innate ability to quickly identify and eliminate abnormal stress antigen expressing cells. And so that core biological behavior, coupled with the ability to edit in and out certain traits, makes this I think, a kind of a great canvas for the tools that Paul and Beam and great companies like MaxCyte are able to offer us. So again, I want to be very clear. Celularity is very much focused on being the provider of the delivery, the platform to deliver these tools and use these tools in as efficient, cost-effective, and clinically relevant manner as possible. So because of that, the placental NK cell, and I guess across the board, natural killer cells in general, makes them really promising candidates for obviously cancer immunotherapy, since that fundamental stress ligand attacking biology of the cell makes it ideal for targeting certain malignancies. But then when you can genetically modify and target tumors with increased specificity, you're dramatically enhancing what nature has created. And that, to me, as a clinician, as a surgeon who's spent the last several decades trying to find a way to turn living cells into biological scalpels, if you will, pardon my pun there, but we think that this capability of the natural capability of NK cells to recognize a range of cancer cells without being prior sensitized to them, and their potential for off-the-shelf allogeneic use and scalability makes them an ideal tool for a range of these therapeutic applications. All that being said, at Celularity, we're very we're avid consumers of the techniques and technologies that companies like Beam are producing, and are very open to how those tools can allow us to refine the behavior of our particular NK cell products. So I want to just say openly, we see ourselves as the beneficiaries of all of that great work in refining these tool sets, and are very open to how using them makes the core technology of NK cells, or T cells for that matter, more useful, safer, and ultimately more scalable and cost effective in the clinical applications.
Thanks, Bob. Okay. Paul, before we move on, I certainly want to give you the opportunity to provide a response as well, perhaps talking about the advantages of NK cells for genetic engineering, but also perhaps some of the challenges from your experience.
Yeah, sure. So we have worked with NK cells as one of the several cell types for base editing delivery, and I'm by no means the cell type expert that Robert is, but I can talk about our experience, which has been that we looked at them as a cell type for their advantages in not having graft versus host disease and being able to be expanded and deliver large doses of cells for an allocell type. They were a cell type that was for us fairly easy to grow, and we were able to bring in house and be able to edit effectively and reach large batches of cells for allo applications. The challenges that we saw, we were working around how to cryopreserve and deliver them as a true pharmaceutical product, and that program is sort of in an early discovery stage. So we're still working out these challenges, but that's the extent of our experience at Beam with NK cells.
Good. Okay. I want to
and to add to that, Aaron, I think Paul put that really, really well. Look, bottom line is that if a primary challenge to modifying NK cells is the lack of gene engineering tools, and it's often difficult to virally transduce NK cells due to the absence of certain receptors on the envelope for the envelope protein like VSVG, then NK cells are really sensitive to some of the other ways of getting the edit into the cell. And I think that's going to improve over time and make the technical operations lead, the manufacturing guy in the cell therapy business, give them a greater sense of artistic license, again, for lack of a better description. So in other words, the more effectively we can edit in very specific ways, the more creative the scientific community in places like Celularity they're going to, hey, this is what we really need. This is going to make the ideal NK cell for targeting this unique tumor type. That's the way I look at it.